No.
I think Tim is actually there, but we cannot see him.
Oh, okay.
Because he's having problems. So wait, he's sending a message. Oh, I am live. Hello, everyone, and welcome to this week's Neurotheory Forum. I'm Everton Agnes. I'm a project leader at the Biozentrum in Basel, Switzerland, University of Basel. And so today, Tim, we had a bit of a technical problem. So that's why we had a bit of a delay, but now everything is fixed. And today I have the pleasure to introduce the speaker, who is Ioana Carcea. She is an assistant professor at Rutgers Brain Health Institute in the US. A bit of background, so she did undergrad in medicine in Romania, and then she moved to the US to do a PhD in neurobiology at the Mount Sinai School of Medicine. And then she did a postdoc with Rob Franke in New York too. And now she has her own lab at the Rutgers Brain Health Institute. And Ioana is most interested in neuromodulators and how they control auditory perception and social behavior. And today she's going to talk about well, I cannot see the title now.
There?
Yeah, social transmission of maternal behavior. And so I'm looking forward to hear her newest work. And without further ado, please, Ioana.
Okay, thank you. Thank you, Everton. I'm gonna start the slides. Is this working fine? Can you see now the full screen slide? Okay, I'll assume that's a yes.
Yes.
Okay, great. Thank you for the introduction, and thank you, Tim, for inviting me to come and virtually speak to all of you today. It's really great to virtually meet you. So today I will maybe start by announcing that my talk is not going to be theoretical. It's basically all of the data I will show, it's experimental data. But I am very hopeful that going forward, I can find collaborators, perhaps among you, who can take this experimental work to the next level into theory. So the project that I will present today, I started it when I was still a postdoc with Rob Franke. Actually, most of the work has actually been done in his lab, and we are now continuing to work on it and wrap it up. And this project concerns the social transmission of maternal behavior in mice. Now, maternal behavior is essential for the survival of most mammalian and avian species. In fact, it is such an important behavior that in many of these species, unrelated conspecifics contribute to taking care of the young. These individuals are called alloparents, or non-biological parents, and they have been so far described in more than 130 different species, including primates, whales, dogs, and rodents. Now, rodents, and in particular mice, represent a great model to study how these alloparenting skills develop in the lab. And that is because when in the wild, the mice, in particular house mice, live in colonies, generally composed of a male, a couple of reproductive females, and several non-reproductive females who usually are daughters of the colony that stick around to help with caring for the next generation of pups. Now, this happens in the wild, and in laboratory settings, we can reproduce this, in part at least, this form of living, and we can also assess maternal behavior in mice quite straightforward. So there is one particular test that we use that's very robust. It is called pup retrieval behavior. What we do here is to isolate a pup from the nest. When isolated, this pup will start emitting distress calls in the ultrasonic range, like the example call that I'm showing here. Let me try to get a laser here. When mothers hear these calls, they immediately, within seconds, locate the pup, pick it up, and retrieve it back into the nest. It has been shown previously that this retrieval behavior depends on the activity of the left auditory cortex, where these pup calls are primarily processed. Now, as I mentioned, mothers do perform this behavior robustly. This is what they do to protect their pups from possible danger and to keep them warm. They retrieve them back into the nest. Interestingly and importantly, virgins that have experience with pups can also perform this behavior. Also, when oxytocin, a hormone which we know to be heavily released in mothers during labor and lactation, when this hormone is given to virgins, either systemically or topically in the left auditory cortex, these animals can also start retrieving pups. On the contrary, inexperienced virgins or animals where the activity of the oxytocin system has been experimentally decreased do not retrieve pups. They do not perform this behavior. So what we can see here is that experience and oxytocin go hand in hand when it comes to pup retrieval behavior and more broadly to maternal behavior in mice. So we wonder then, is it possible that experience uses the endogenous oxytocin system in order to induce this maternal behavior in virgin female mice? Before I go into how we started addressing this question, I just want to tell you more about what I mean by experience. So traditionally, virgins were allowed to gain experience with pups by co-housing them with a number of pups for hours or days. More recently, we started doing co-housing not just with pups, but also with the mother of those pups, which is also called a dam. So first off, we wanted to understand whether there is a difference between these two different experiences in terms of how quickly and how robustly virgins can acquire pup retrieval behavior. So what I'm showing you here in this graph, we plotted the probability that virgins retrieve pups out of 10 trials over time, over days of co-housing. And what you can see here in these empty symbols is the behavior of virgins when they are co-housed only with pups. And you can see that they can acquire pup retrieval behavior over time. However, when we co-house virgins with mother and pups, we see that this acquisition is much accelerated. Virgins start retrieving pups much sooner when they are co-housed with the mother and pups. So we then wondered, how is this socially mediated acquisition of pup retrieval behavior implemented? How does it happen? So we addressed this question at 3 levels. First, we looked at behavior. We were particularly interested to determine if there are relevant interactions between mother and virgin that could accelerate the acquisition of pup retrieval behavior in virgins. Secondly, we were interested in whether neuromodulatory systems, in particular the oxytocin system, which we know to be the major player in maternal behavior, whether it is involved in or it is activated by the behaviors that we identified during co-housing. And lastly, we wanted to know whether co-housing behaviors can induce plasticity for pup call representations in the left auditory cortex, which, as you remember, is essential for this pup retrieval behavior. So to address these questions, we build a system for continuously monitoring co-housing behavior over the course of days. So this system has a camera for video taping and a microphone, an ultrasonic microphone for recording pup vocalizations and also adult vocalizations if they happen, and a neural activity recording system in order to record the activity of oxytocin neurons in the virgin's hypothalamus. So with this system, we acquired days of video recordings in this co-housing settings. And initially, at least, we manually analyzed this, looking for interactions between mother and virgins that might be relevant for the acquisition of pup retrieval behavior in virgins. And we identified two behaviors that we find to be relevant. The first one is this one where the dam seems to be directing the virgin towards the nest. I'll show you an example in the video here. So what you see in the corner of the co-housing enclosure is the nest with pups inside, and the mother, the dam, is in this snapshot already emerging out of the nest, just coming out of the nest. The virgin is located in a different part of the co-housing cage, and you will see the mother going towards her and chasing her towards the nest. So there's the mother grabbing the virgin and directing her towards the nest. We started calling this behavior shepherding behavior because this is really what it most resembles. So we wanted to first look at the spatial distribution of this behavior. Here I'm showing you an example of the co-housing enclosure. The white circle here represents the location of the nest, and the yellow pixels represent the probability that this shepherding will start at that particular location in the cage. As you can see, shepherding can start pretty much anywhere in the cage, except for the nest. The nest is pretty empty. However, when we look at where shepherding ends, we see that very consistently the shepherding ends within the nest. So the red pixels here represent the probability of this behavior ending. So there really seems to be this spatial distribution from outside of the, or direction of the behavior, from outside of the nest towards the nest. Importantly, we believe that this is a motivated behavior, in part because we don't see it happening quite as often when there are no pups in the nest. So the mother performs this shepherding behavior very frequently when there's pups in the nest, but not when the nest is empty. So what are the behavior consequences of this shepherding behavior on the virgin? One of the consequences is pretty clear and kind of expected. The virgin starts spending more and more time in the nest as the co-housing progresses. Here you see the percent of total time that virgin is in the nest, and you can see that it goes up over time. But another consequence that was surprising is that shepherding also correlates with the probability that virgins start retrieving at the end of day one. So here what you see is the probability of retrieval in virgins at the end of day one, and on the x-axis you see the frequency of shepherding per hour during that first day of co-housing. And you can see a very robust correlation between the two. So in terms of the shepherding behavior, what I showed you is that dams produce this behavior. About 85% of all the chasings that mothers do are towards the nest, so we can call them shepherding. This behavior depends on the presence of pups in the nest. It is not as frequent when the nest is empty, and it results in virgins spending progressively more time with pups and also retrieving pups more often. The other behavior that I wanted to introduce is the fact that mothers self-generate retrievals during co-housing. So what we observe is that oftentimes the mothers, when exiting the nest, accidentally drag one or two pups with them that are still suckling. And when she moves out of the nest, the pups are dropped, and then she returns to retrieve those pups. And again, I'm going to show you an example of that behavior here. You see the mother in the nest. She came out and accidentally dragged the pup with her, and then she returns to retrieve the pup back into the nest. And all this time, the virgin was in the nest and had the possibility to observe what the mother is doing. And these virgins have this possibility, this exposure to maternal retrievals repeatedly during co-housing, on average once every 2 hours or so. So we wondered then whether this experience, being exposed to maternal retrievals, can induce pup retrieval behavior in virgins. So one way to look at this is again to look at the correlation between retrieval probability at the end of first day of co-housing and the frequency of them retrievals during that first co-housing day. And we can see that these are correlated, not as robustly as we've seen with shepherding. However, there is a correlation there. But moreover, we then took this maternal retrieval or this behavior outside of the co-housing context. And now we repeatedly expose virgins to them retrievals without having them co-housed at all. So we just have the virgin observe 10 them retrievals, and then we give her a break, and we test pup retrieval behavior in the virgin. And we do this for 4 consecutive days. And we wondered whether virgins will be able to retrieve pups themselves after being exposed to maternal retrievals. Here I'm showing you data from 15 virgins, where we're plotting in green the probability that virgin retrieves after the first, second, third, or fourth day of this experiment. As you can see here, many of the virgins start retrieving soon after the start of the experiment, day 1 and some on day 2. Overall, about 11 out of 15 animals retrieved after being exposed to them retrievals in this paradigm. But then we wondered, what is the modality, or what are the modalities by which the information from the mother is conveyed to the virgin to help her acquire this pup retrieval behavior? So the first thing we tried was to block any touch interaction, any touch between virgin and either pups or mother. So to do this, we placed a transparent barrier in the cage that separates the them and pups from the virgin, physically separates them, but allows visual, auditory, and most likely olfactory information to go across the barrier. I'll show you an example of this experiment here, how it was conducted. You see here is the partition of the mother and pups, and this is the partition of the virgin. We isolated the pup, and now we're waiting for the mother to retrieve it. The virgin was right at the barrier. She had the possibility to observe what the mother was doing. And then we test behavior in the virgin to see if she retrieves pups, and we see in this example that she can immediately retrieve pups. So in this paradigm with the transparent barrier, again, we see that the majority of virgins that we tested out of 19 were capable of retrieving pups after being exposed to maternal retrievals. However, if we now make the barrier opaque, blocking visual information, we no longer see an acquisition of this pup retrieval behavior in virgins. In fact, only 2 out of 18 animals retrieved after undergoing this paradigm. So there seems to be visually guided social learning of pup retrieval behavior in virgins. And this resembles, in a way, behaviors that have been described previously in many different species, where observers can acquire a behavior from a demonstrator. That's been shown in fear learning, also in mice, also in birds during vocal learning, and interestingly, even in species that are asocial, like octopuses, they can also learn from each other. Okay, so we described 2 behaviors, shepherding and social learning, that can contribute to the acquisition of pup retrieval behavior by the co-housed virgins. Now, let's look to see whether these behaviors can engage the oxytocin neuromodulatory system in order to induce these changes in behavior. So, to start this line of the experiments, we first wanted to see whether the activity of oxytocin cells in the paraventricular nucleus of the hypothalamus, or PVN, are important or are necessary for the acquisition of pup retrieval behavior. So what we did here, we induced the transduction of chemogenetic virus, an inhibitory designer receptor exclusively activated by designer drugs. We induced its transduction specifically in oxytocin neurons using a transgenic mouse. And then in slice recordings, we confirmed that we can silence these oxytocin neurons by applying the designer drug called CNO, named CNO. So here you can see the activity of an example oxytocin cells before, this is slice recordings, this is before the application of CNO, and this is after CNO was applied to the slice. So we can successfully silence these neurons. Now let's see, if we do this during behavior, how does this silencing affect the acquisition of pup retrieval behavior? So here I'm showing you the success of pup retrieval behavior in experimental and control animals. What we can see here is that when oxytocin neurons are silenced using this chemogenetic approach, the success of pup retrieval behavior decreases, or the acquisition of pup retrieval behavior decreases, indicating that the firing of oxytocin neurons is important for the acquisition of maternal care in virgins. So then we wanted to see how the behaviors we identified during co-housing impact the activity of oxytocin neurons in PVN, if they do so at all. So for this, we did single unit recordings in the paraventricular nucleus of the hypothalamus, which is very small and deep in the brain. So we wanted to confirm the proper location of our electrode bundle in this region by doing co-registered computer tomograph and structured MRI imaging. So here you see an example of an electrode in red, correctly implanted in the paraventricular nucleus. Moreover, in order to specifically record the activity of oxytocin cells in this brain region, we used optical tagged recordings. We induced the transduction of channelrhodopsin, specifically in oxytocin cells in PVN. And then we used blue light delivered from an optic fiber to induce the activity of all cells expressing, or of cells expressing channelrhodopsin. So here you see an example recording in this paradigm, you see that the cells responds very robustly and with short latency to the onset of the blue light. And because of this type of response, we can conclude that this recorded neuron is a bona fide oxytocin neuron. So with these tools in hand, we then recorded activity in PVN throughout the co-housing procedure. And we aligned the activity of neurons to the onset of the behaviors that we are interested in. So here you see an example cell recorded in PVN. The spike trains are aligned to the onset of shepherding. The shepherding episodes are highlighted in red. And as you can expect from any spontaneous behavior, they are quite different in terms of their duration and also their frequency. So to determine whether this behavior can change the activity, the firing of this neuron, we compared the firing rate during this behavior episodes with a baseline that immediately precedes the behavior. And what you can see is that this example cell is activated robustly by shepherding. Overall, we find that about 90% or about 90% of oxytocin cells are activated by shepherding, and only 12%, about 12% of other unidentified PVN neurons can be activated by shepherding. Interestingly, neurons that do significantly respond to shepherding do not respond or are not activated by a behavior that's similar in the motor pattern, but it's quite different in the social meaning. So here what I'm showing you is that when the virgin chases the mother, these neurons that are activated by shepherding are no longer activated, indicating that the motor action is not the factor that induces the increase in activity for these neurons. I mentioned that shepherding generally ends by with the virgin entering the nest. So we also wanted to look at that particular time point in the behavior. What happens in terms of neuronal activity when the virgin enters the nest? So here I'm showing you nine units recorded simultaneously in PVN. One of these units, shown in blue here, is a bona fide oxytocin cell. And what you can see is that both the oxytocin neuron and some of the unidentified PVN neurons seem to be activated by nest entry. And that's more clearly shown here, for example, cells in the peristimulus time histograms. So overall, we find that a similar percentage of neurons, of oxytocin and unidentified PVN cells respond to nest entry. So about 32-33% of the cells are activated when the virgin enters the nest. Importantly, the cells are activated when the entry into the nest follows shepherding, but also when the entry is voluntary. So this would indicate that perhaps there are sensory cues associated with the nest. They could be visual, tactile, olfactory, and so on, that induce this increase in the activity of PVN cells. What about experiencing DEM retrievals? So here again, I'm showing you 4 units simultaneously recorded in the virgin PVN. The unit shown in blue here is a bona fide oxytocin cell. The spike trends are aligned to the moment when the mother starts retrieving pups. Preceding that moment, there's an interval of time when the pup that is isolated accidentally produces pup calls. So you can see an example of pup call there. So what we observe is that the oxytocin cell, as well as other unidentified PVN neurons, can respond to this experience of observing the mother produce pup retrieval. So about 30% of oxytocin cells in the virgin PVN can respond to what the mother is doing, to the retrieval that the mother executes, and only about 17% of unidentified PVN neurons respond to this behavior. So there are several questions, two that we were particularly interested in that come out, or that are, we started asking them after looking at this data in more detail. And one of these questions is whether the population of neurons that are activated by these different behaviors is the same or different. So what I'm showing you here is that there's little overlap between the population of neurons that respond to nest entry in gray here, shepherding in red, dem retrieval in blue. There's in fact only a small percentage of neurons that respond to two of these behaviors. And this is true for oxytocin cells, and it is also true for unidentified PVN neurons. In this case, we also find cells that appear to respond to all three co-housing behaviors that we considered. Also, another question that we had from looking at this data was whether there might be some synchrony in the activity of these neurons in PVN. So what we did here to look at the coactive pairs of neurons, we did the dot product between the frequency or the firing frequency of pairs of neurons. And we find that pairs that include at least one oxytocin neurons seem to be more coactive, or actually more precisely, there's more coactive pairs if the pair includes an oxytocin neuron. And that's true for both shepherding behavior, nest entry, and dem retrieval behavior. Okay, so what I showed you in this part of the talk was that shepherding, nest entry, and observing pup retrieval can activate PVN oxytocin neurons and other neurons in PVN. Now, I should also mention that other behaviors that we observe during co-housing are not capable of activating these neurons to the same extent or not at all. So, for example, if the virgin enters an empty nest without pups, we do not see this activation of oxytocin and other PVN neurons. Okay, so now that we see that there are behaviors that can specifically activate oxytocin cells in PVN, the next question would be whether this can be used as a mechanism to induce cortical plasticity for pup calls, and eventually this would lead to pup retrieval behavior. So previous data from Rob Fromke's lab showed that there is a difference in how the auditory cortex in dams and the naive virgins responds to played-back pup calls. In dams, the auditory cortex is very responsive to this played-back pup calls, whereas in virgins, we don't see much activity. So this difference in activity seems to be important for the execution of pup retrieval behavior. When the auditory cortex in dams is suppressed, or the activity in this region is suppressed with muscimol, we see a decrease in pup retrieval behavior compared to the pre-inactivation and post-inactivation conditions. And more recently, more recent work from Rob Fromke's lab by Jennifer Schiavo et al. showed that during co-housing, we can observe a transition between this low responsiveness to high responsiveness state in the virgin auditory cortex. And we also found this transition in single unit recordings in the auditory cortex, as well as fiber photometry recordings in this region. So again, we see an increase in responses to playback pup calls. It seems to be peaking at the time when the virgin starts retrieving pups. So to understand whether this transition can occur due to the oxytocin neuron activity elicited by the behaviors we described during co-housing, we did dual recordings. We recorded calcium signals, so fiber photometry recordings in the auditory cortex, and single unit recordings in the virgin PVN. And then we had these dually implanted virgins exposed to maternal retrievals across the transparent barrier. And what we observed is that there is a correlation between the activity of PVN neurons and the activity of the auditory cortex. And this is shown here. It is a robust correlation. So we wondered if then oxytocin being released by this interactions during co-housing, if it can modulate responses to pup calls in the auditory cortex, we would expect that inhibiting the oxytocin action in the auditory cortex might prevent the acquisition of pup retrieval behavior. And so again, we tested this hypothesis in the transparent barrier paradigm for the pup retrieval exposure. And what we did here was to infuse right before exposure to demo retrievals, we infused the oxytocin receptor antagonist in the auditory cortex. And then when we tested retrieval in virgins, what we observed is that compared to the saline infusions, when we applied the oxytocin receptor inhibitor, virgins were not as capable of acquiring pup retrieval behavior anymore. So we see here that only 2 out of 10 animals that received this antagonist before exposures to demo retrievals were not capable of acquiring the behavior. Or only 2 were capable of acquiring, the rest were not capable. Furthermore, we then wanted to determine if we can replace the visual stimulus by stimulating the activity of oxytocin fibers that project into the auditory cortex and still induce the behavior. So here, we have the opaque barrier condition. Usually in this paradigm, the virgin is not capable of acquiring pup retrieval behavior. However, if we stimulate oxytocin fibers in the auditory cortex, despite the virgin not observing what's happening on the other side of the barrier, she will be capable of acquiring pup retrieval behavior. So you can see here, quite surprisingly, that 5 out of 5 virgins can acquire this behavior in this paradigm. So what I showed you then in the last part of the talk was that oxytocin can modulate cortical representation of pup calls to enable pup retrieval behavior in virgins. Does this go like really fast, or okay? I think we're good in terms of timing. Okay, so I would like to end by acknowledging, first of all, Rob from here, whom I work with, and where I started doing this project, and I did most of the project in his lab, in very close collaboration with people in his lab. Some of them are listed here, but really not all of them. I'm very grateful to Rumi, who has been my main collaborator on this project when I was both in Rob's lab and now she's in my lab. I also want to acknowledge people in my lab at Rutgers and collaborators at Rutgers and other extramural collaborators. I would also like to thank my funding sources for their support, and we are interested in hiring talented postdocs. If anybody is interested, please contact me. Thank you.
Thank you, Anna. It's really nice talk.
Thank you.
So people, if you have any question in the audience, please, you can write them in the chat box, or there is an ask a question box below. Please use, we have a lot of time now. And also after, so before I start questions, after the talk, we are going to have a Zoom chat with the speaker, and I'm posting the link in the chat. So please join us for further discussions afterwards. So I have one question now related to the circuit in this, related to this pup retrieval and this acquiring this maternity aspect. So it's clear that the oxytocin somehow connects to the auditory cortex for the release of oxytocin or the neurons. But what drives these neurons? And clearly you said, so if you block the visual aspect, the animals don't learn anymore. So I was wondering if there is any pathway from visual system, or there is something there that could hint on it.
Thank you. This is a really wonderful question, and we have very fresh data that might be able to answer some of that. I'm not showing it here, but basically what we did retrograde tracing using rabies viruses from oxytocin neurons in PVN in order to identify what brain structures can send projections to this neurons, so sending inputs to these neurons. And we did find that a structure that functions in the visual system, the superior colliculus, does send projections to these oxytocin cells. So it is possible that the visual information, before reaching the visual cortex, really, at subcortical levels, is transmitted to oxytocin cells. So we don't know yet if these neurons are excitatory, probably because they're projection neurons, they're long-range projection neurons. So it's likely that they're excitatory cells, but we don't know that yet. It is possible that visual information can activate oxytocin cells via this pathway.
So it makes sense then with the behavior that you see that the circuit somehow resembles what you see.
Yes, yes.
So we have questions here. Let's see if I can ask the Robert Scholz to join us to ask the question. Let me see, how do I do this? Okay, so there is a technical question problem here. I cannot figure out how to call him on the screen. So I'm going to ask the question. So Robert Scholz is asking if you think the olfactory /sound or olfactory or sound cues play a role in the barrier condition when using oxytocin to induce maternal behavior related plasticity.
Yeah, absolutely. I do think that that's likely to happen as well. Mice are very much guided by their olfactory modality in pretty much all the behaviors that they do, and so, or a lot of them. So I would be surprised if the olfactory information was not involved in this, but it remains to be determined exactly what pathway in terms of the neural pathway from olfactory areas to oxytocin cells might transmit this information, and exactly what olfactory information would be important. Is it the smell of pups? Is there something particular about this pups, perhaps pheromones that they release that could induce the activity of oxytocin cells? So that part I haven't started exploring yet. I think it's fascinating. I also think it's very complex and probably not as easy to address, but I do think it's a fascinating story or question, and I do expect olfactory information to play a major role in this.
Thank you. We have Peter Jonas here also to ask a question, please. Oh, you're muted. Wait.
Now it should work.
Oh, yeah.
Yeah.
Thank you very much. Nice talk.
Thank you.
Very nice recordings from this small area, which presumably is very difficult. I have two questions. First, bursting. So do the PV neurons burst abundantly because that's a general requirement for peptide release, presumably also oxytocin release?
Yes, I haven't looked much at bursting, but what I can say is that okay, there is a possibility that other neurotransmitters might be released from these cells when they are active, and not just oxytocin. And perhaps these other neurotransmitters, maybe glutamate or other things, could play an important role in this plasticity of the auditory cortex. However, because we did that experiment where we blocked the activity of the receptor in the auditory cortex, of the oxytocin receptor in the auditory cortex, we have reasons to believe that oxytocin is also released when these neurons are active, and that it also plays an important role. I haven't tested directly glutamate transmission to see whether it also might be required for the acquisition of this behavior, but we know that oxytocin is needed. Now, does this happen on, every time the virgin sees the mother retrieving? Are we going to get a burst that is capable of releasing oxytocin? Perhaps not, but perhaps it is sufficient to have this happen, 2, 3, 4 out of 10 trials in order for the plasticity to occur.
But you would see this in the unit recordings, right?
Yes, I would see this. And yeah, I haven't looked specifically at bursting, but there are neurons that fire. When we see the increase in firing, there are neurons that fire up to 30 hertz, which would be the firing frequency where we know it's the optimal, that is optimal for the release of oxytocin.
And the second short question, you have already alluded to the inputs of the PV cells.
Yes.
What about what exactly are the target cells? Are these principal neurons or also interneurons involved? Or does a lot of the effect go over release of oxytocin into the blood circulation?
So this is also a fascinating issue, and I don't know, but I don't believe that the blood oxytocin will be able to have much of an effect in virgins. It probably mostly acts, when in the blood, oxytocin mostly acts on peripheral organs. It cannot cross the blood-brain barrier very easily to act centrally. So I do believe that most of the actions that we see in behavior are going to be driven by effects of oxytocin centrally. And that can happen either by being released at nerve terminals that project to different parts of the brain, like the cortex, the amygdala. We see very nice projections to the amygdala from these neurons. But it's interesting, we and others have seen sort of a little bit of a decoupling when it comes to receptor expression and projections from oxytocin neurons. Okay, so there are more regions that express the receptor than regions where we see oxytocin cells projecting to. And so what I think happens, there's a volume transmission, and I'm not just me, but like there's a thought in the field that there is a volume transmission. Oxytocin might increase in the cerebrospinal fluid, and then might, or just might increase in the parenchyma and diffuse from the site of release and act on multiple regions without necessarily having this matched, axon, oxytocin axon, and receptor on the other side. Now, when it comes to actions on circuits, yes, oxytocin can modulate the activity of both inhibitory and excitatory neurons. It does seem to play a role in this excitatory-inhibitory balance, and work from previous work from Rob's lab showed that very nicely in a paper by Marlin et al., where they did intracellular recordings in vivo in the auditory cortex, and they looked at inhibitory and excitatory inputs and how they are modulated by oxytocin.
Great, thank you.
Thank you.
Hi, fascinating talk. I have two questions. One is, if there is more than one virgin observing this behavior, is there some sort of a favoritism as to which of the two virgins gets recruited? And the second question is this sort of transferable? Can a virgin teach another virgin this pup retrieval mechanism? Can it sort of extend further on? Can we have open pups?
Yeah, that's wonderful.
Yeah.
We haven't done that experiment to look at sort of chain transmission of behavior, but I would think that that's possible. We don't know for sure, though. Again, going back to the olfactory question, right? There could be that there's a convergence of inputs needed in order to induce the activity of oxytocin cells and therefore induce the behavior. So perhaps in addition to the visual stimulus, which we think would be the same between observing a mother or observing a virgin, there might be an olfactory signal from the mother that's specific to her physiological state. She's lactating, she's gonna probably put out different pheromones than a virgin. So if this convergence of inputs is necessary, then perhaps we will not see this transmission of behavior. But it's very interesting. I haven't done that experiment. Now I'm curious to do it and see. And the other question you had, if two virgins are watching the behavior, what determines who's learning and who's not learning?
Exactly. Who gets recruited into this? Is there some sort of
also a very, very good question. We do see that some learn and some don't learn, and I didn't show you the data, but when we look at the activity of oxytocin cells in learners and non-learners, we see a difference. So oxytocin cells get activated only in learners. So I would think that the difference then has to be, somewhere before the activation of oxytocin cells, right? So something must determine why in some animals oxytocin neurons can respond to this experience and in others not. I don't know what the difference could be. It could be the menstrual cycle. We did not really control for that in these experiments. So that might be a factor. I think it would be important for us to determine that. Perhaps animals are more receptive in estrus or whatnot. But I don't know for sure. What I can tell you is that it's not at the level of, we don't think it's at the level of the auditory cortex, right? So on that leg of the circuit, we don't think it's happening there. It's probably something that happens before oxytocin cells get activated.
Thank you.
Thank you. Great questions.
So we have a few more questions before going to the Zoom meeting. So one of them from Bayar Menza, so he's not coming to screen, is do mothers increase their pup retrieval behavior when there is a virgin present to teach?
Huh. Fascinating. So basically, is there instruction? Is there, is this animal motivated to instruct another animal to do this behavior? We don't know that. I guess we can, in our paradigm, mothers retrieve reliably, whether somebody observes them or not, meaning that they retrieve almost always 10 out of 10 trials. But there might be differences in the latency with which they retrieve that could be different between the two conditions. And I think it'll be very interesting to look at that, to see if they retrieve faster when they're observed. Yeah, it's quite fascinating. I'm very curious to know that too. But we haven't done that experiment. And like the fact that the mothers, when co-housed, they drag pups out. In my opinion, that's accidental. I don't think the dragging out is motivated. I don't think it's intentional. However, in some mothers, occasionally we see another way in which pups can get out of the nest, and that is the mothers start digging in the nest and just literally push the pups out with their hind legs, and then they go and retrieve them. So that's, to me, seems more like an intentional behavior, but who knows what the intention of an animal is. But it's just just like anecdotally, I guess.
This happens more often when there is a virgin watching or present.
Yeah, we should look at that. I don't know the answer to that, but we should definitely look at that.
Cool.
Yeah.
So there is a question also from Nila. There is no last name here, but she's asking, there seems a considerable number of virgins that never express pup retrieval in various of your experiments. Do they differ in any way behaviorally from other virgins? And have you followed those into motherhood?
Oh, no, I haven't followed any of these in the motherhood. I think, in fact, one of our reviewers asked us the same thing. I think it's a fascinating question, and something that definitely should be done to see whether this, evolutionarily indeed is advantageous for the species, right? I think it would be. I think, generally what we see is that once an animal retrieves, it will retrieve constantly. So it would probably prepare the animal for motherhood, right? Once they have their own pups, they would already know how to do this. But we haven't actually looked at this with real data to be able to tell for sure. The first question was that some learn to retrieve and some don't. Are there other behavior differences? I haven't noticed signs of stress being different between this virgins that learn and those that don't learn. I mean, it's possible. We didn't really look at physiological measures of stress, but like behavior measures, we didn't notice any particular clear difference. If anything, maybe sometimes it's different than what I would expect. I would expect that when animals are stressed out, they don't learn, right? But in fact, sometimes I see that the virgins that are more agitated are the ones that start retrieving faster. But it's not a conclusive thing one way or another. We haven't done the classical test for anxiety, like the open field test or elevated plus maze or anything like that. We don't know if there is a difference, or perhaps another factor that could contribute is the ranking in the social hierarchy. We also didn't look at that yet to see if it's a factor.
Oh, okay, and there is another one from Adriana Mansafo. So she's asking, do virgins ever have a tendency to nest with the dam and pups on their own, or they are mostly forced by the dam?
At least initially, they don't nest with the mother and pups. So it seems like they need this sort of help, from the mother to start entering the nest by themselves. They do eventually do that. They do enter the nest voluntarily, but it just doesn't seem to be something that happens right away.
Okay. And so last one before we go to our Zoom meeting from Tim. So he's also saying that he has a broken computer, so he cannot come online today. But so the question is, did you have plans to also look at the effect of trauma on the retrieval behavior? And if so, diminished if diminished behavior can be recused by way of activating oxytocin cells?
So like a stressful event, trauma, is it?
Yeah, if you can induce trauma, yeah.
I do have plans to look at this. I don't know if trauma would be exactly how I would describe it, but I do intend to look at the effects of threat and other environmental stressors like temperature to see whether these behaviors can be modulated up and down by this environmental factors. I was talking to students and postdocs before this, and I was saying that, in my opinion, all these social behaviors are just, or can be summarized as adaptive mechanisms to the environment. And so I would expect that changes in the environment will change these behaviors. Specifically how, we started looking at temperature. Zara, one of the students in the lab, is, she's having some great data looking at how shepherding behavior produced by the mother changes with temperature. And that's, hopefully we'll talk about that another time. But also threat is another important factor that I'm interested in. I do think threat might increase these types of behavior because they would constitute part of the tend-and-befriend response that's been described in people and some animals in response to threat. So, we have the classical fight or flight response that's perhaps prevalent in males. We don't know that for sure. But it seems that in females, in particular, the response to threat is quite different. And it is this tendency to take care of your offsprings, to recruit a network of peers or conspecifics to basically protect you from the threat. Whether you want to get lost in the crowd, and that's like one way in which you can protect yourself, or who knows, in humans, we share information. Gossip is a way of protecting ourselves by sharing information. And that's a response to threat. So I would expect trauma, threat to increase these behaviors.
Yeah. But then if you could, so Tim is complementing the question. So you could break this retrieval behavior, and then so the question that comes after, if you can rescue it. So if you do something to break this type of behavior, if you can rescue it afterwards.
I think it depends where we break it, right? So if we remove oxytocin cells, if we kill oxytocin cells in the virgin brain, will we be able to rescue the behavior with other mechanisms? I think it depends. It depends. If we eliminate oxytocin cells early in development, I'm pretty sure there's going to be some compensation. As I said, this is an essential behavior. Maternal behavior is essential. I'm pretty sure the brain has multiple mechanisms to take care of it, to implement it, even when one of them goes down. So, serotonin is another neuromodulator that has been shown to play important roles in social behavior in general. It might be recruited for this maternal behavior as well, or dopamine. I'm not sure yet, but I think if the oxytocin cells are removed later on in life, it might be harder to rescue the behavior. Yeah, but it's interesting. I don't know. I think something that can be addressed.
Yeah. So I think, so those were all the questions that we had. And now we can move to our...
Okay. Thank you all.
Yeah, thank you very much, Ioana.
Thank you.
Yeah, thank you very much.
Bye.
It was a really interesting talk. And so anyone who wants to continue discussions or ask any other question to Ioana, or just pass by to say hi, please join us. The link is on the chat, and we are going there now. Thank you again, Ioana.
Thank you.